Our research of the past 2 decades has been early diagnosis of diabetes complications. In that sense, we looked at many early events that happen in the eye, and one of the recent projects was about how diabetic cataracts form. And to understand how they form, we had to go back to the earliest stages called pathogenesis, how the very beginning starts. And right there, something very curious happened. When we saw the very first lesions, which we refer to as microlesions, hyperreflective microlesions that nobody had seen before in the animal eyes. When we saw them, in half of the animals, the blood sugar was not above normal yet. They did become diabetic later, but at the time when we saw the microlesions, the blood sugar was within the normal range. A lot of our research is based on imaging. And as Richard Feynman said, many fundamental principles of biology are very easy to explore. One has to just look at them. The difficulty, of course, lies in how to look at them. You need equipment, you need technologies, which at different times grow. And our colleagues at Hoch have knowledge of know-how, how to go about imaging the eye in ways that were not possible before.
We at the Hoch were responsible to detect the microlesions in the animal's lens. This was very challenging because we need a very high resolution to detect this tiny microlesions in the lens on one hand. On the other hand, we need a high depth of field. And for this we use a special surgical microscope which has superior optical properties compared to a standard microscope.
Now that we know that the pathogenesis starts before blood glucose becomes abnormally high, that also meant that there is a different mechanism which nobody had explored before. To go about it, we developed new histological techniques where we could see areas around the lens, particularly the ciliary body and the zonules that hold the lens in place. And when we did that, we found that immune cells migrate from the ciliary bodies along the zonules towards the lens. Some of the immune cells seem to be penetrating the lens capsule, and in those areas, the epithelial cells that line up the inner surface of the capsule change their properties. Some of them died, some of them proliferated, and some of them started to migrate. Now, these are all new mechanistic findings which seem to have other causes than previously thought. We now know where to go and look for cause and effect and new ways of preventing, perhaps treating these early findings.
We at the Hoch were responsible for the optical part of this project. We had some major challenges with the rat lens. On one hand, we need a high resolution to detect these microlesions, and on the other hand, we need a high depth of focus. With a standard slit lamp ophthalmologists use normally in clinical routine, we are not able to detect these microlesions in the lens. Therefore, we used a surgical microscope with a dual illumination, a high depth of focus and a high resolution. And the depth of focus of our surgical microscope fits perfect to the axial length of the rat's eye.
If you go back in the literature, the current understanding of how diabetic cataracts form relies on a hypothesis called the sugar cataract hypothesis. It was coined over half a century ago, and the essence of it is that when glucose levels are high, through conversion in a pathway called the polio pathway, glucose turns into a sugar alcohol molecule called sorbitol, And the sorbitol has osmotic properties, holds water molecules to it, which leads to opaqueness of the lens. The fibers swell and the lens becomes cataracts. Now, this hypothesis necessitates that there is high levels of glucose. Our finding that the glucose was normal in the lens and in the blood of half of the animals suggested that there is something else going on. And when data contradicts an existing theory, that's room for scientific discovery.
We want to investigate these deposits in more detail to look for the optical behavior and the size, and then we have to look for the right image modality for the clinical routine. Here we used a surgical microscope, but I think for a routine clinical diagnostic, the optical coherence tomography will be the perfect tool. In the next project, we investigate the use of OCT or the other image modalities for a clinical use of this new insights.
Now we learn that actual tissue damage happens before the conventional definition of diabetes starts. So there must be something else going on in the pre-diabetic stage that triggers changes, and once we understand what the molecules are that cause those changes, then this is new opportunity for developing novel treatments. But at the same time, if there is a tangible lesion that you can image in the eye of an individual with diabetes, then this can be used in a non-invasive manner for screening, broad-based screening. That's of tremendous value. The world has currently over 500 million individuals with diabetes, the great majority of which are type 2 diabetic, and being able to screen for incipient lesions is a great plus to the individual, but also to the public health. Especially in the brain, it would be of high clinical value to understand the earliest lesion that happened, perhaps in the pre-diabetic state, and whether we can provide mechanistic insights that makes it easier to find them. We are interested in also looking in the same animals in which we found these lesions in other tissues and see whether other organs are affected as we found lesions in the lens. My laboratory has been in long-term collaboration with Christoph Rußmann's laboratory in Göttingen, and we have the optimal situation where our biology benefits from the technologies that his laboratory is specialized to develop. At this point, we look forward to continuing our transatlantic collaboration between my lab in Boston and Christoph Rußmann's lab in Göttingen. Our most recent projects have been awarded a grant by the National Science Foundation in cooperation with the German Ministry for Education and Research, the MBF, for a transatlantic collaboration.